All right, in this lesson, we're going to be practicing the ability to identify mutations. So I already have the assignment open. I just want to kind of walk through how you are to complete this assignment. So um, our goal for this assignment is to be able to identify mutations that happen in DNA and then also what effects they have on the outcome of the amino acids that are produced. So we've been learning about transcription and translation and how DNA is put into action into making proteins. And remember, proteins are made by stringing amino acids together on a chain. So we're going to go look at how mutations in DNA might affect which amino acids are added to a chain, which would then possibly affect the protein that gets made. So this assignment is actually a worksheet. And here we are in the assignment, as I mentioned before. And there's a Word document. So if you scroll down, you'll see the Word document is attached down here. And you're going to be completing this similar to the way we practiced it last week when we were translating the code in mRNA into amino acids. So we are going to use this chart here. We definitely want to be able to use that to help us identify the correct amino acids. So we're going to go ahead and open this up. Now what happens when sometimes when you open this, it might open up inside of Schoology and there'll be a downloads button you'll want to hit. But for me, it happened to just download. So you can see it down here in the bottom in the downloads bar. And I'm just going to go ahead and click this up arrow and say open. Really, you can just click right on the file name and it should open up in Word as well. All right, so it should look like this. If you see a bar across the top that says that it's in read-only mode or that you need to click a little link in order to edit the file, you want to go ahead and click that link or the button that says Enable Editing. And then it'll be in this mode here that will allow you to work inside this document. But since we need to see this document and our code translation chart at the same time, here's what we can do. So if you want to just click and drag your Word document, you should know how to do that. Click and drag it over to the edge, and it's actually going to fill up half of the screen. And if you're on Windows 10, Windows 10 is going to want to know, well, what do you want on the other half of your screen? So it shows you all of your windows, and you're going to pick the one that has the chart in it. So now we can easily see our Word document as well as our codon chart, so we can do the translation that we need to do. All right, so here we have some sequences, and this here is the original DNA sequence, and this is going to be kind of the thing you keep coming back to as you go through each of the mutations below and fill them out. The chart's available down on the second page as well, just in case you need it. But setting it up this way with the web browser on one side of the screen and the Word document on the other should work just fine. So you can see that the original DNA sequence is here, and the bases are separated into groups of 3, which means they're separated into their codons for you. Kind of did that in advance to make things a little easier. And then in the second line here in the green, which is already filled out for you, is the mRNA sequence that would result from transcription inside of the nucleus. Now keep in mind, when you come across an A in the original DNA sequence, the actual um, the actual uh, the actual base is going to be U. Okay, so instead of T, it's going to be U. So the mRNA sequence is going to be AUG, So T goes to A goes to U, C pairs with G. And then in the next codon, the next three bases, we're going to have A matched with U, C matched with G, C matched with G, and then T matched with A, T matched with A, G matched with C, and you just keep going down the line like that, which you should know how to do. All right, once we have the mRNA sequence, we can then figure out what amino acid that each one of those codons will code for. So if we go to our wheel over here, we have AUG as our first codon, so we're going to start with A, then go to U, and then go to G, and that brings us to methionine. I always say that wrong. It's methionine. So we can just use the first three letters or the first syllable. You can actually use the first syllable to be even a little more specific, because some of the amino acids kind of have the same first couple letters. But anyway, in this case, we get methionine, and so we're going to put MET or METH here as our amino acid sequence in this row. UGG goes here. UGG gives us tryptophan. So we're going to put in TRY or TRYP. Either one is fine. You see that that's already done here. AAC gives us ASP. Put ASP underneath our AAC. And then the CGC codes for, what is that? CGC codes for arginine. So we're going to put ARG in there. You see that's already in there. UGC codes for cysteine. So we're going to put CYS as our amino acid there. And then UGA codes for a stop codon. So we're going to just write in or type in the word stop. So that's where the ribose knows, hey, my work here is done. I can stop. Okay. Now we have, if we go to the next box down, which is blue, we're going to compare this mutated DNA sequence to our original DNA sequence to figure out where that mutation occurs. So the first thing we want to do is look for a change. So let's see, right now TAC matches with TAC, that's all good. ACC matches up with no it doesn't, look at that, there's a difference. So let's see, it's ACC, but down here it's ATC. So here's where the mutation occurs from the original, and this is our changed codon. So we want to highlight those. We're going to do that in every mutated DNA sequence. We're going to highlight the changes when we find them. It's going to help you to kind of see how that change, that mutation, affects the protein that's supposed to be built by this set of instructions. All right, let's keep going. TTG matches up both in the green and the blue. GCG is the same. ACG and ACG is good. ACT and ACT is good. Okay, so our next step is to, again, translate. No, I'm sorry, we're going to transcribe into an mRNA sequence. All right, so TAC is going to be AUG, and ATC, which is different than what we had before, that's going to be, all right, so A matches with U, so that's going to be U, and the T is going to be an A, and the C is going to be a G, so it's UAG, and then TTG becomes AAC, GCG becomes CGC, ACG is UGC, and ACT codes for stop. Okay. So where's the change? So our mRNA sequence should be the same too, except for we've got this mutation. So clearly the UGG is now a UAG. So let's go ahead and highlight that as well. Oh, what happened? AC, that should be GC, and stop. Okay, I don't know why it keeps double underlining. I think that's Word trying to correct my spelling or grammar or something. All right, our amino acid sequence is going to start out the same with that typical start codon, met or meth. I like met better. It doesn't sound like it's an illegal drug. And then UAG is different than what we had before. So we can't just copy our amino acid sequence from above. We have to figure this out. So we go to our chart. UAG is stop. So this is a stop codon. So what the heck does that mean? That means the ribose stops right there. We don't have to list the rest of the amino acids because the ribose would stop at that point and would not bring in any of the other amino acids. So the next row asks us, will there likely be effects? And we can say yes. What are the effects? That's the next question. What are they? The effects are the amino acid chain stops. And no protein is made. And what kind of a mutation is this? So we know there are two main types of mutations, point mutations and frame shift mutations. And if we compare our mutated DNA sequence number 1 to our original DNA sequence, we see that just one base is changed throughout the entire sequence. Everything else remains the same. Just that one base gets changed. So this is a point mutation because it's just one point in the sequence that got changed. So we can put that down here. Point mutation. And you could even be more specific if you like. You can put it's a substitution. I don't know how to spell. Substitution mutation because a thymine was substituted for a cytosine in that DNA sequence inadvertently. So that is a substitution type point mutation. Now, that's how you fill out one of these boxes. So I've done the first one for you. So you definitely want to fill this out the way that I did. And then you're going to carry on doing the translation of the mRNA sequence. I'm sorry, that is a transcription. And then you're going to translate it into an amino acid sequence and determine whether or not there's any effects. Don't forget to highlight any of the mutations that you find and identify them. So you have 3 more boxes to go, and each time you're going to compare this mutated sequence to your original sequence. Okay, so once you filled out this blue box, the mutated DNA sequence number 1, you're not going to use it anymore. Once you've completed the DNA sequence number 2, you're not going to use it anymore. You're going to go to number 3 and compare it again to the green box at the top, the original DNA sequence. So we're always comparing a mutated DNA sequence to our original DNA sequence. Never would we want to compare a mutated DNA sequence to another mutated DNA sequence. Okay, so the color coding should help you with that. Green is the original, blue is what you're working on. Okay, so finish the other 3, and then you're going to go ahead and upload your finished Word document. So make sure you save it. Make sure you put your name on it up here, right here, so that I can see your name when I open the file. And get that uploaded, and I'll be able to check out your work and make sure that you've got a clear understanding of this so that I know that you're ready for the quiz.